Okay, so you will notice after the last module, which was Chapter 8, Skin Disorders, we are skipping Chapter 9 on musculoskeletal disorders. And the reason we are skipping it is because almost all of the other classes in this program are concerned with musculoskeletal disorders. And so we're not going to cover it in this class because it will be covered in many of your other classes. So we're going to go on to Chapter 10, which is blood and circulatory system disorders. So the circulatory system consists of the cardiovascular system and the lymphatic system. So in Chapter 10, we'll talk about cardiovascular system, and in Chapter 11, we'll talk about the lymphatic system. So in this chapter, Chapter 10, we're going to cover blood vessels, blood, and associated disorders. Okay? So in the interest of chunking these lectures up into small lectures, first I was going to get all crazy about blood through the whole, through a really long lecture, but I'm going to try to divide it up a little bit. So this PowerPoint is going to be divided up into 2 lectures. So here's our anatomy of major arteries. So all of the systems in the human body are interrelated, and they have to interact in order to maintain homeostasis. But all of the different systems are dependent on blood. Blood circulates the body and transports oxygen and nutrient to the tissues. It removes waste products of cellular metabolism, and it plays a critical role in the immune defenses, as well as maintaining body homeostasis. So I like to think of the arteries and veins as being the information superhighway of the body, or the superhighway anyway. They're the fluid transport system of the body. The nervous system is sort of the information transport, and they all depend on each other. So blood and lymph, and blood is an essential body fluid, lymph is another essential body fluid. They're transported throughout the body through a complex system of vessels and the pumping action of the heart. So there are different features in the circulation of blood and the circulation of lymph, and we will talk about all of those different characteristics in the next 2 chapters. So arteries, capillaries, and veins are a closed system for the distribution of blood throughout the body. Even though things can get in and out at the capillary area, like oxygen and nutrients and waste products, and fluids, it's still a closed system. And when you look at this, and these are on pages 186 and 187 in your book, Major Arteries and Major Veins, you'll see that they are paired. Just like most of the other systems in our body, like we have 2 sets of our skeletal, our major skeletal components. We have left and right of our nervous system components, and we also have left and right of our vascular components. So, of course, the central ones, we only have 1 heart, we only have 1 aorta. So, remembering, of course, that there are 2 separate circulations. The pulmonary circulation, which allows the exchange of oxygen and carbon dioxide in the lungs, and the systemic circulation, which provides for the exchange of nutrients and waste throughout the body, and all the cells throughout the body. So I love these microscopic pictures. I think they're super cool. But the arteries transport blood away from the heart and into the lungs or into the body tissues. And arterioles are their smaller branches that control the amount of blood flowing into the capillaries. So you can't just dump all the blood from the huge arteries into the capillaries without overflowing them. So it starts getting smaller and smaller to control that amount of blood. And the arterioles control that through a degree of contraction of smooth muscle in the vessel walls, either vasoconstriction or vasodilation, depending on how much blood you need in a given area. So we talked about that with inflammatory processes, and this is how it works. So capillaries are super tiny, small vessels organized in networks throughout the microcirculation of the body. So the blood flows really slowly through the capillaries, and there are precapillary sphincters that determine the amount of blood that flow from the arterioles into the individual capillaries, depending on the metabolic needs of the tissues. Remember how when we were talking about inflammation, we talked about how an inflammatory response can open up unused capillary beds? This is how the control is exerted. So those chemical messengers are going to cause vasodilation or vasoconstriction that are going to facilitate this. So you can think of the veins and the arteries as being the highways and the capillaries as being the surface streets. So you have to drive more slowly on a surface street. You can't drive at highway speeds. So the blood flow slows down through the capillaries. Then small venules collect blood from the capillary beds and start moving it back towards the heart. The larger veins collect blood draining from the venules, and normally a high percentage of the blood, about 70%, is in the veins at any one time. So the veins are called capacitance vessels. They hold the fluid. The blood flow in the veins is dependent on skeletal muscle action, respiratory movements, and gravity. So in our larger veins, we have valves. So the graphic there on the left shows vein versus artery. In the veins, we have valves. And the valves play an important role in keeping the blood flowing towards the heart. It doesn't allow backflow. So when you do have problems with the valves, it's considered venous insufficiency. You do get backflow and pooling of the blood. So respiratory movements and muscle movements assist the movement of blood through the trunk. So when we talked about the consequences of immobility, it changes your blood flow. So immobility is bad. That's why in physical therapy we are concerned with mobility. We want to mobilize people. So arteries go to arterioles to transport blood away from the heart. Venules go to veins to return blood back to the heart. Capillaries are the little guys, microcirculation within the tissues. They're the surface streets. The systemic circulation exchanges gases, nutrients, and wastes in the tissues. Pulmonary circulation exchanges gases in the lungs, primarily oxygen and carbon dioxide. Here's our little mini system, if you want to look at it, and we'll talk about the different layers of the blood vessels. So, arteries and veins are made up of 3 layers. The tunica means layer. Tunica intima, the inside layer, it's the endothelial layer. Tunica media is a layer of smooth muscle that controls the diameter and lumen size of the blood vessel. That's the middle layer. And the tunica externa or tunica adventitia is the outer connective tissue layer that contains elastin and collagen fibers. That allows that elasticity, that moving, and it's collagen type 1 fibers. So there are actually tiny blood vessels called the vasa vasorum that supply blood to the tissues of the vessel wall itself. Isn't that cool? So the large arteries have to be super elastic to adjust to changes in blood volume that occur during the cardiac cycle. So in the genetic chapter, when we were talking about Marfan syndrome, spontaneous dissection of the aortic artery is common because it affects connective tissue. So the connective tissues in the walls of the vessels themselves have to be flexible. So the aorta can expand during systole to prevent systolic pressure from rising too high, and during diastole the walls have to recoil to maintain adequate diastolic pressure. So the veins have thinner walls than arteries and less smooth muscle. That's how you can distinguish them in these micrographs. In the bottom picture here, the one with the big red circle, which is a lot of smooth muscle, is the artery, and a little bit less of the red circle is the vein. And you can see in the upper picture on the right that big ring of smooth muscle tissue around the artery and a slightly smaller ring of smooth muscle tissue around the vein. And then outside of it, there's more muscle tissue, and that helps with the pumping effect. Okay. So, localized vasodilation or vasoconstriction in arterioles is a reflex adjustment in a small area of a tissue or organ, and it varies depending on the needs of the cells in the area. So if you have a decrease in pH or an increase in carbon dioxide or a decrease in oxygen, it leads to local vasodilation to help get more blood into that area. Chemical mediators such as histamine can cause that as well. So the local changes don't affect the systemic blood pressure. They just affect that one little area. Some of our hormones and neurotransmitters, norepinephrine and epinephrine, increase systemic vasoconstriction. And some of our cardiac hormones, angiotensin, also act as vasoconstrictors. So all of those are subject to treatment by medications because you can affect the levels of those in your blood. So we have our vascular tone or vasomotor tone is maintained by the sympathetic nervous system. And we'll talk way more about this in neuro, but really the sympathetic nervous system's biggest job, besides the fight or flight stuff that we've always talked about, is to maintain the optimum blood flow in the body. So you get partial vasoconstriction throughout the body to ensure that you have continued circulation of blood. So capillary walls are thinner, they just have that one endothelial layer, and that facilitates the exchange of fluids and everything else that needs to exchange. So kind of cool. Cool stuff. So blood, here we are talking about blood. It's our major transport pathway for oxygen and nutrients to all tissues. It removes waste products of cellular metabolism, and it plays a critical role in the body's defenses and immune system, and it helps to maintain homeostasis. So it provides a mechanism for maintaining and controlling body temperature, distributing heat through the peripheral tissues. It's the medium through which body fluid levels and blood pressure are measured and adjusted by hormones and neural controls. So there are clotting factors circulating the blood that are readily available if they're needed. And we have buffer systems in our blood, which we talked about in the chapter about fluid and electrolyte and acid-base balance, to maintain that stable pH that has to be in that super narrow range to keep us from, death. So that's a good thing. So blood, we have about 5 liters of blood as adults in our body, which is, a decent amount of blood. Blood consists of water and dissolved solutes, which make up about 55% of the whole blood volume. The remaining 45% is composed of cells or formed liquid elements. So erythrocytes are red blood cells, leukocytes are white blood cells, and thrombocytes or platelets are clotting cells, if you want to call them. So in this micrograph, let's find a bigger picture of it. There it is. The little donut ones are the erythrocytes. The big blobby ones are the leukocytes, and the itty bitty ones are the platelets or the thrombocytes. And we'll go back. Okay. So the hematocrit refers to the proportion of cells, which is largely made up of erythrocytes, in the blood, and indicates the viscosity of the blood. In general, adult male humans have a higher hematocrit than adult female humans. An elevated hematocrit could indicate dehydration because there's less fluid, therefore a higher proportion is cells. A low hematocrit might result from blood loss or anemia. So let's just paint a little scenario here. You are a PTA, you are working with someone in the hospital. Let's just say he's an adult male, and he just had surgery. And you're a smart PTA, so you check his labs before you go work with him. You check it out, and he's got a hematocrit of 30. So he has fewer red blood cells to circulate oxygen. You think he might be dizzy when he gets up? I think he might be. And it's likely that that low hematocrit resulted from blood loss during his surgery. So you want to take care to check his O2 sats. You want to make sure that he's got adequate perfusion of oxygenated blood. If not, you're going to put him on oxygen because we can do that. You are also going to check in with him while he's exercising to make sure that he's not feeling dizzy, because nothing's worse than having your patient faint. Well, there are things worse, but let's just say that's a bad thing. So the average hematocrit for adult males is 42 to 52%, and for adult females is 37 to 48%. So look at your patient's hematocrit. Get an idea of what they're likely to be like. If it's super low, you might have to do bed exercise. Maybe you're not getting them out of bed today if they have a super low hematocrit. If you're unsure about that, check with the nurse. The nurse is a fine pipeline of information for you. The plasma is the clear yellowish fluid that remains after you remove the cells. So normally in the lab, they spin the tubes of blood in a centrifuge to concentrate the cells at the bottom and the plasma at the top, and then they draw off the plasma to test it. Serum is the fluid and solutes that remain after the cells and fibrinogen have been removed from the plasma. And usually they do that chemically in the lab. The plasma proteins include albumin, which maintains osmotic pressure in the blood, globulins or antibodies, and fibrinogen, which is essential for clotting. So this table is on page 189 in the book. It's an awesome table. It's a really good one to know. So the fluid components of blood are not just there to give the cellular components a way to get around. They also have a lot of function. So as far as the cellular components go, the white blood cells, they act as phagocytes. They respond to an allergic response. They release histamines and heparin involved in inflammatory responses. They are involved in cell-mediated immunity and humoral immunity, the T and B lymphocytes. And the monocytes also turn into macrophages and perform phagocytosis when there's tissue damage. Erythrocytes, hemoglobin transports oxygen. That is their thing. Thrombocytes, blood clotting. Okay, in the fluid and chemical components of blood, albumin is the big dissolved protein which maintains the osmotic pressure of blood. And we'll talk about that when we talk about the lymphedema, how that changes during injury or illness. So globulins are the antibodies circulating in blood. Fibrinogen is a blood clotting factor, and we'll talk about hemostasis and how that works. Water is a medium for the transport and exchange of body fluids and secretions. We also have a lot of things involved in cellular metabolism in the chemical components of blood. Amino acids, carbohydrates, and lipids, otherwise known as protein, carbohydrates, and fats. Those are our macronutrients. Then we have vitamins, hormones, enzymes, electrolytes, and wastes. So lots of stuff in the fluid component of blood. So all blood cells originate from the red bone marrow. And in adults, the red bone marrow is found in flat and irregular bones, ribs, vertebrae, sternum, and pelvis. Pretty neat, right? The iliac crest is in the pelvic bone is a common site for bone marrow aspiration for biopsy or for bone marrow donation, if you're going to donate bone marrow to someone. So that's pretty cool. Various blood cells develop from a single stem cell, and that single stem cell is called a pluripotential hematopoietic stem cell. If that's not a great name, I don't know what is. On page, this is the little development diagram from the book on page 190, starting with that pluripotential hematopoietic stem cell. During the process of hemopoiesis or hematopoiesis, The basic cell differentiates into committed stem cell types for each type of blood cell. So that first one is the original stem cell. The 5 on the next layer are committed types of stem cells for the different types of blood cells. So each type of committed stem cell proliferates and matures depending on the needs of the body. So any pathological condition of blood that refers to disorders involving cellular components of blood is called a dyscrasia. So there are, we're going to talk about some specific blood dyscrasias, and the way I remember it is like, this is crazy. It's a disorder concerning blood cells. It's a dyscrasia. So we're going to talk about the different types of cells. Red blood cells, or erythrocytes, RBCs are their abbreviation. They're biconcave flexible discs. They look like donuts, but with thin centers rather than holes. Or if you're a bagel fan, they look like Bialys. So they're flat in the middle and round on the sides. So they're non-nucleated when they're mature, and they contain hemoglobin. That's their job. The size and structure of those red blood cells is essential for easy passage through small capillaries. Red blood cell production is stimulated by the hormone erythropoietin, which originates from the kidney. So the kidney creates erythropoietin. It stimulates erythrocyte production in the red bone marrow, and it's a response to tissue hypoxia, or insufficient oxygen available to cells. So red blood cells normally constitute most of the cell volume in blood. Adequate red blood cell production and maturation depend on the availability of many raw materials, including amino acids, iron, vitamin B12, vitamin B6, and folic acid. So hemoglobin itself consists of the globin portion, which is the protein portion, 2 pairs of amino acid chains, and 4 heme groups. And the heme groups contain that ferrous iron atom to which the oxygen molecules attach. The heme, which is the iron, provides the red color associated with oxygen in the lungs. So oxyhemoglobin is bright red, and that's how you can distinguish arterial blood from venous blood. So as the blood circulates through the body, the oxygen dissociates from the hemoglobin, and deoxygenated hemoglobin is dark or bluish red in color, and that's what's found in venous blood. Normally, carbon dioxide, there's another binding site on globin, on hemoglobin, that can carry a little bit of carbon dioxide, but a lot of it is transported as bicarbonate ion in the buffer system in the blood. So carbon monoxide, which you don't want to inhale, bumps oxygen right off its binding site in hemoglobin, and it binds tight and doesn't let go. So that's why it kills you. You get fatal hypoxia from a deficit of oxygen, because the carbon dioxide will not let go of that hemoglobin binding site. When someone has been poisoned by carbon monoxide, their lips are a cherry red color. The lips and face. So that's how you can tell. And it's because of the way it affects the hemoglobin. So normally a red blood cell, its lifespan is about 120 days. As it ages, the cell becomes more rigid, so it can't squeeze through those capillaries as easily. Finally, it gets phagocytized in the spleen or liver and is broken down into, the hemoglobin is broken down into globin and heme, which is then further broken down into amino acids, which are recycled and used again. The iron can be returned to the bone marrow and liver to also be recycled and used again to make more hemoglobin. So excess iron is sometimes stored in the liver, blood, and other tissues. There's a genetic disorder, a hemochromatosis, which is iron overload, basically. And it results in accumulations of the storage form of iron, which can damage the organs. So the balance of the heme component, when it's broken down, is converted to bilirubin, and transported by the blood to the liver, where it is conjugated with another molecule to turn it into bile. And so excessive hemolysis, or destruction of red blood cells, can cause elevated serum bilirubin levels, which result in jaundice, which is the yellow color of the sclera of the eye or the skin. So those can certainly be indicative of liver problems or other blood problems. So the erythropoietin that's produced in the kidney, if people artificially inject erythropoietin into their blood, it will create more red blood cells, and that is what's known in the sporting world as blood doping. So in cycling or whatever sport, it's an advantage to have more red blood cells, and so people inject erythropoietin to create more red blood cells. And it's detectable by having a higher number of red blood cells. But and people have gotten nailed on it in the Tour de France when they tested them afterwards. So they're sneaky about it, though. I'm of the opinion that everybody does it and some people get caught. So here's our blood cells. And there are those white blood cells in there, which are, you know, pretty cool little guys. They do a lot for us. They're only about 1% of our blood volume, as opposed to our red blood cells, which are a much higher percentage. There are 3 types, granulocytes and 2 different types of agranulocytes. They all develop and differentiate from an original stem cell in the bone marrow. Unlike the erythrocytes, which are stimulated by hypoxia in the tissues, which stimulates the erythropoietin in the kidneys, or the kidneys to produce that, leukocytes are stimulated by what's called colony-stimulating factors, CSFs, produced by macrophages and T lymphocytes. So sometimes certain ones are produced, certain chemical messengers are produced to increase certain types of white blood cells during an inflammatory response. So that is important, because you need those white blood cells for the immune response and the inflammatory response. So there are 5 types of white blood cells. Lymphocytes, making up 30 to 40% of white blood cells. And those include the T and B lymphocytes in the immune response. Neutrophils, which are the largest component, comprising 50 to 60%. There are a lot of them, but they only survive 4 days, and they're our first response on phagocytosis. Remember, they're the non-specific responders. If you see increased immature neutrophils, that can be indicative of a bacterial infection. Basophils migrate from the blood and enter the tissue to become mast cells that release histamine and heparin. They can be fixed in certain tissues, or they can just wander around looking for something to do. The eosinophils, they tend to combat the effects of histamine, and they're increased when you are having an allergic reaction or a parasitic infection. Pretty interesting, huh? Monocytes, they enter the tissue and become macrophages, are big eaters. They act as phagocytes when tissue damage occurs. So when they do a differential count, what they're doing is they're counting the different types of the different white blood cells. And different imbalances in them can indicate infections or allergic reactions. Thrombocytes, also called platelets, they are an essential part of the blood clotting process, also known as hemostasis. So thrombocytes are not really cells, they're really tiny, itty-bitty little non-nucleated fragments from larger cells, which are called megakaryocytes. Platelets stick to damaged tissue as well as to each other, and they form a temporary plug that seals small breaks in blood vessels. Or they can adhere to rough surfaces and foreign material. So there's a neat chart in the book, which is not this chart. It's this chart, okay. It's on page 192. That shows how anticoagulants work, which is kind of neat. But we're going to talk about the 3 steps of hemostasis. So first, the immediate response of blood vessels to an injury is vasoconstriction. And in small blood vessels, it decreases blood flow and may allow a platelet plug to form. Then the thrombocytes adhere to the underlying tissue at the site of injury, and if the blood vessel is small, the platelet plug can take care of it. If it's not, blood clotting or coagulation mechanism is required in larger vessels, and it involves the clotting factors that are present in inactive forms in the circulating blood, and they're activated through a sequence of reactions. And the reactions are listed on this diagram. So the clot formation, it's listed, 1 through 5 in the on page 193. It's a cascade of events. It's a sequence of events. So damaged tissue and platelets release factors that stimulate a series of reactions. Prothrombin activator is usually the first factor produced. PTA, we might remember that one. Prothrombin, also known as factor 2, is usually inactive in the plasma, and it's converted to thrombin. Thrombin has a lot of different functions in the blood. Fibrinogen, which is known as factor 1, and I don't know why they're not in order, just to make me crazy, I think. It's converted into fibrin through the action of thrombin. A fibrin mesh forms to trap cells and make up a solid clot or thrombus, stopping the flow of blood. The clot gradually shrinks or retracts, pulling the edges of the damaged tissue closer together and sealing the site. So the circulating clotting factors are produced primarily in the liver, and their numbers relate to the order in which they were discovered. They didn't just number them out of order to make me crazy, but they discovered fibrinogen first, and then they discovered prothrombin. So a person who is lacking one of these factors or some other component of the blood clotting cascade has hemophilia, and we will talk more about hemophilia. So vitamin K, which is a fat-soluble vitamin, is required for the synthesis of most clotting factors. Calcium ions are also essential for many steps in the clotting process. Vitamin K is available in your favorite leafy green vegetables. I prefer chard and spinach. Some people like kale. Take your pick. I think I'll have some chard for dinner tonight. That sounds good. So there are other measures you can take, which you know from first aid, hopefully, to facilitate the clotting process: applying pressure and cold, which is a vasoconstrictor, reducing blood flow in the area. And they also can apply a thrombin solution directly to speed up clotting, which is kind of cool. So as usual, there is a delicate balance between hemostasis and anticoagulation. So if you have too much coagulation, then you form clots, and that can cause you to have a stroke or heart attack. If you have too little coagulation, you can bleed to death. So just like a lot of homeostatic processes, there is a balance. So you don't want to form clots unnecessarily and cause infarctions. So sometimes they put you on anticoagulant medications. Okay, in the next section, we are going to talk about blood typing.